Thank you very much. I think my conclusions are going to be very much the same as Inga's were. What I will try to do is fill in some of the details along the way, and I'm going to focus more on the chemosensory side than the brain side. So I'm interested in these gut sensory cells because of their involvement in metabolism. So I come from the field of diabetes and obesity, and many of these gut hormones actually, we know they're involved in the coordination of nutrient absorption and digestion. But they're also involved in other processes that control metabolism. So actually gut motility, I think, is very important in how the postprandial profiles of plasma nutrients actually rise, because depending on how the food is moving down the gut and being absorbed, you'll get different plasma profiles of glucose if you've had a glucose drink. But many of the hormones are involved in insulin secretion, so GIP and GLP-1 particularly are very strong stimuli of insulin secretion. If you don't have GLP-1 and GIP, you actually produce about half the amount of insulin release. And they're involved in the control of appetite. So gut hormones actually, we think, are very important potential new treatments for diabetes and obesity. And GLP-1, as you'll all know, is already used to treat type 2 diabetes because of its stimulation on insulin secretion and its suppression of appetite. So after you eat a meal, the nutrients, how am I going to control this? So the nutrients pass down the gut lumen and they make contact with these chemosensory cells in the epithelium of the gut. And you have the different hormones that differentially expressed down the length of the GI tract. So here I've got representation of GIP that is more found proximally, GLP-1 that is found some of it proximally, but quite a lot of it more distally, right down to the rectum, and PYY, which actually is a bit more distal. So if you measure concentrations of GLP-1, GIP, and PYY in people who've had a meal, you get slightly different profiles. GIP goes up about 10-fold, we found. GLP-1 goes up about 2-fold, and PYY goes up about 20 to 50%. And I think this is because when you eat the meal, most of the cells producing GIP actually make contact with the absorbed nutrients. So most of the cells producing GIP will be stimulated, so you get a big signal to the background noise. Only a proportion of the GLP-1 producing cells will make contact with the nutrients, so you get a smaller signal against the background, and many of the PYY cells won't even see the absorbed nutrients, so they give you the background signal here, and then you just see the PYY, the nutrient-induced signal on top of that. But the focus of our lab actually is to try to work out how is it that the digested food actually is absorbed and sensed by the chemosensory mechanisms on these cells. And particularly I will talk to you about cells producing GLP-1, but I actually think that the cells producing GLP-1, GIP, CCK, they're actually part, I think, of a very broad continuum. And all the studies to date have suggested that the mechanisms on one are very similar to the mechanisms on another. So to study this, we started off using a cell line called glutag, which is also fairly similar to the STC1 cells that we've heard a little bit about. We decided that cell lines weren't going to tell us everything, and we started making transgenic mouse models where we've expressed fluorescent sensors in enterocrine cells. So this is one where the L cells producing GLP-1 are labelled with a venous, a YFP derivative, and you can see it then if you do a primary culture of the intestine. You can see the fluorescent cells, and then we do single-cell electrophysiology and imaging of cyclic AMP and calcium and things to see if we can work out how these cells actually respond when you add different nutrients. And we've labelled a number of the different chemosensory types by targeting the hormonal promoters to drive the transgene expression. And then more recently, now we're starting to move on instead of, so for these primary intestinal preparations is killing a mouse once a week and doing a preparation. We now do quite a lot of it in organoids, so we've made organoids from the transgenic mouse lines, and here you can see GCAMP3 co-localizing with GLP-1 in organoids, so we can then keep these going in culture and preserve having to use mice all the time. This is one of the early studies showing that L cells are electrically active via action potentials which are nutrient dependent. I think this recording was actually from a glutag cell, but we've got very similar recordings from primary L cells from, the next ones we got were from colon. Now we can get them from ileal organoids. So I think this is a genuine property of L cells, that they fire these action potentials that look quite like neuronal ones. They overshoot, and I'm not going to show you the data, but they are dependent on sodium and calcium, voltage-dependent ion channels. And they depolarize in response to several nutrients, and I'll show you some of the data of how we think that that is happening. And that is the real question, is how is it that the digested nutrients turn into signals that end up resulting in triggering of electrical activity, calcium entry, which is mostly L-type triggering secretion, and this downstream signaling pathways. And this is my conclusion, just so you know where we're heading, which is we think that there are that the enterocrine cells harness nutrient transporters on the apical membrane. So there are sodium-coupled transporters, the one that Inga mentioned, that I'll tell you a bit more about, the SGLT1, and there are proton-coupled peptide transporters, and we think that this sets up a membrane depolarization responsible for detection of some nutrients. But other nutrients are detected by these G-protein coupled receptors, and some of them are GQ coupled and some of them are GS coupled, and I'll give you an example of each in a minute. So this is a summary that I think looks rather like Inga's summary, showing actually that the cells respond to a wide range of digested macronutrients, as well as cinnamon, mustard, bile acids, short-chain fatty acids, other microbial products. And they do it via a number of different transporters and ion channels and G-protein coupled receptors. And we've also found some responses to, some pathways responsive to AVP and angiotensin, so they can also detect some basolateral signaling and galanin receptors. And the ones I'm going to tell you a bit more about are sugar sensing, long-chain fatty acid sensing, and bile acid sensing, just to get an overall picture. So the glucose sensing, Inga's already told you what my conclusion is, but the sugar sensing by triggering GLP-1 secretion was found that it needed sodium, and it needed sugars that were transportable, and it could be blocked by inhibitors of glucose uptake, so fluoridazine, which blocks SGLT1. That doesn't actually tell you how it's happening, it tells you what the dependence is. And it could have several possible explanations. It could be that the sugars need to get across, they need to be absorbed by the brush border, because they need to target receptors on the other side. And so those could be sweet taste receptors or other pathways. It could be that the sugars need to get through the brush border because they need to get inside the cells, and then they'll be detected once they're inside the cell. Or it could be that the sugar sensing is directly linked to the transport mechanism itself. So I'm going to take you through those three different alternatives and tell you why I believe the third one. The first reason to say actually that we don't think that it's sugar just needing to get across the brush border to get access to the basolateral side is that if you give people glucose by mouth and then a similar matched glucose load intravenously, only the glucose by mouth works and intravenous glucose doesn't. This is an in vitro experiment, however, where we've done these primary cultures that I showed you the picture of. We've applied glucose. You can stimulate GLP-1 secretion when you add glucose, but if you don't have SGLT1 there, the glucose doesn't trigger secretion. And in that case, it's not because glucose hasn't got to this side, because we haven't got a polarized epithelium here. We're applying glucose that gets to both sides. So it's something to do with the transport, not just that it needs to access that basolateral side. This is just a brief interlude to show that we don't think that the chemosensory glucose sensor on the L cells is the sweet taste receptor. This is microarray expression of receptors in L cells compared with non-L cells. So we've purified the cells by FACS, and then we did microarray expression analysis. So anything in this green triangle is something that is significantly more expressed in an L cell than a neighboring cell. And here you can see the two, the TAS1R2, TAS1R3 that make up the sweet taste receptor. and they're similarly expressed in L cells compared with the neighbouring cells, and actually anything that's less than 100 on this is actually fairly poorly expressed. So we don't think that this particular glucose sensing mechanism is the sweet taste receptor, and we also couldn't trigger secretion from these primary cultures with sucralose and acesulfame K, which we would have predicted if it was a purely sweet taste. So I don't think that what we're looking at here is sweet taste. Could it be that you just need to get sugars inside the cell and then there's another sensory pathway happening within the cell? So glucose enters across the apical membrane by SGLT, and then there's the diffusive equilibration across the basolateral membrane by GLUT transporters, mostly GLUT2. So we used inhibitors of SGLT1, which is fluoridazine, or of GLUT, we used fluoretin. And we did fluorescent FRET imaging of the glucose concentration inside the cell using a FRET-based glucose sensor. So the sensor gives you an increased FRET signal when the cytoplasmic glucose concentration goes up. So here you can see we added glucose to the outside of the cells and the concentration went up inside the cells. When we blocked the GLUT with fluoretin, we no longer saw a glucose rise inside the cell. When we blocked SGLT with fluoridazine, the glucose concentration still went up. However, if we do the secretion experiments in parallel, fluoridazine blocks the glucose-triggered secretion and fluoretin doesn't. So we here have a complete dissociation between fluoretin, which blocks glucose uptake but doesn't block secretion, and fluoridazine, which blocks secretion here but actually didn't block the glucose signal. So we think it's not the glucose concentration inside the cell that's important, it's something physically to do with the SGLT1 itself. So this is a picture showing the SGLT1 on a small intestinal villus and a venous labelled L cell. And these transporters, when they take every glucose molecule that goes through them, they take 2 sodium ions. So they actually set up a small inward current driven by the sodium ions. And we think we can record that small current, and it's just enough to depolarize the cells. Here you can see that glucose stimulates a calcium rise in wild-type L cells, but if you knock out SGLT1, you don't get that calcium signal. And we think that is actually this very small depolarizing current actually entering with the SGLT1 that's setting up a small membrane depolarization that you can see electrophysiology, and then that triggers these action potentials linked to the calcium entry and secretion. So if that works for SGLT1, could it work also for other similar transporters that also take ions in with their substrates on the brush border? And we have some evidence for it in glutag cells with amino acids. There's a sodium-coupled uptake mechanism, and also for the proton-coupled peptide transporter. So we think the cells can sense glucose and probably some sensing of amino acids and dipeptides via this substrate uptake transporters. I think for the proteins, I don't think it's all this. I think, I'm not going to show you, but also the calcium sensing receptor that Inga mentioned, I think is actually very important for amino acid sensing, but I think this is probably part of it. So what about bile acids? We were thinking along the same mechanism for bile acids. And bile acids actually do 2 things when you apply them to cells. One is, this is L cells. One is that they elevate cyclic AMP. So this is measured using another FRET-based sensor that changes its FRET when the cyclic AMP concentrations go up. So this is taurolithicolic acid, and you can see it elevates cyclic AMP concentrations. You can also see that this is taurodeoxycholic acid, increases calcium concentrations in L cells, and this is measured using GCAMP expressed in the cells. So we did wonder whether this increase in calcium triggered by taurodeoxycholic acid could be an electrogenic action on the apical sodium-coupled bile acid transporter. We think this cyclic AMP rise is due to GP bar, as I'll tell you a bit more about. But this ASBT takes sodium with bile acids, and we thought it could be responsible for this. It's expressed in L cells, as well as in the neighbouring cells, terminal ileum, but we were recording this in terminal ileum. However, we got an ASBT inhibitor from GSK and applied it in these calcium imaging studies, and actually it didn't in any way block the effect of TDCA. So I think, actually, having thought about this harder, that these substrate transporters will act as nutrient sensors, but you have to have a fairly high substrate flux in order to generate a big enough current to trigger the cells. And I think that for bile acids, I think the flux rate is actually just too small to generate a big enough signal. And I don't have any explanation for how this TDCA is triggering the calcium, but we do think that the effect on the cyclic AMP is via this G-protein-coupled bile acid receptor that elevates cyclic AMP. And actually, we set up an Ussing chamber system, firstly, mostly actually to test whether the ASBT inhibitor was actually functional in our system, because we couldn't be sure that it was working at all. So here we sandwich a bit of interstitial tissue between two chambers of an Ussing chamber system, and we've modified it slightly to low volumes so that we can sample from the basolateral membrane and measure GLP-1 concentrations. And here you can see that compared with basal, which is this dashed line at the bottom, bile acids, and I haven't got a GP bioagonist here, but bile acids stimulate the release of GLP-1 into the basolateral compartment. But when we did this with tissue from a GP-bar1 knockout mouse, you could see that the bile acids didn't work anymore. So we think this is a GP-bar signaling pathway. And we showed that the ASBT inhibitor that we got actually worked, because if we applied bile acids to the apical side, they stimulated GLP-1 secretion, but if we applied the ASBT inhibitor, they didn't trigger secretion anymore, whereas it didn't matter if you applied the inhibitor when you applied the bile acids to the basolateral membrane. But the other side of, the other interpretation of this actually is that the bile acids need to get across the basolateral side in order to work. And what we think is required is actually that the bile acid receptor is actually on the basolateral membrane, and that you need to get absorption across the epithelium before it targets the receptors. And I'm going to move swiftly on to long-chain fatty acid detection, because I think this also involves absorption. So in the small intestine, long-chain fatty acids and monoacyl glycerols are reformed back into chylomicrons in the enterocytes to be secreted onto the basolateral side. And there's some quite interesting data that is not from us, but has said that if you block chylomicron production, you can block fat-triggered GIP and CCK secretion. So we wondered about the role of chylomicrons in triggering GLP-1 secretion. And perhaps that you need to get the chylomicrons to get everything across the basolateral side, where it would be detected by FFAR1, that's also GPR40 on the basolateral side. And some infusion studies done in Copenhagen has showed that agonists of FFAR1 only work if you apply them from the vascular side, not the basolateral side. So we found that if we applied chylomicrons to glutag cells, here we could stimulate GLP-1 secretion. And what we did, we thought, well, maybe the chylomicrons need to be lipoprotein lipase, lipolysed, what's the right word, in order to have this effect. So we knocked down LPL, that was lipoprotein lipase, that was highly expressed in glutag cells. And we found that if we knocked out LPL, we lost chylomicron signaling. And then we decided if the LPL is releasing free fatty acids, maybe that's then working through the free fatty acid receptor. So we also knocked down FFAR1, or GPR40, and here you can see that also abolished the chylomicron-triggered GLP-1 secretion, as well as a response to an agonist of GPR40. So we think actually, at least in the small intestine, that the mechanism for long-chain fatty acid detection requires the long-chain fatty acids to be formed into chylomicrons. Now, I don't actually know how much free long-chain fatty acids are released on their own, but the chylomicrons, once they're on this side, we think the free fatty acids can be released again by LPL, and then the long-chain fatty acids target the free fatty acid receptor. They were for working together with the bile acids. And so we wondered, so how are the free fatty acids actually working when they got onto the cells? And we think that they're working via recruitment of this TRP channel, TRPC3. So TAC875 is an agonist of FFAR1, and you can see it depolarizes L cells. This is an L cell from an organoid. And here you can see that TAC875 induces an inward current in these L cells that is blocked by PIRE3, which is an inhibitor of, a fairly specific inhibitor of TRPC3. We also found that TAC875 triggered GLP-1 secretion was blocked in the blue bars here by this same TRPC3 inhibitor. So I actually think that downstream of FFAR1, quite a lot of the mechanism is via GQ-mediated activation of TRPC3, which is then setting up depolarization, and then you get downstream to this same L-type calcium channel opening. So the conclusion of all this, actually, I think, forget about the mechanisms if you want to, but is that we've looked at glucose, which has to be absorbed via SGLT1. We've looked at bile acids, which have to be absorbed to trigger the bile acid receptor on the basolateral side. And we've looked at long-chain fatty acids, which have to be absorbed to reach the free fatty acid receptor. And I think it actually sets up a fairly important principle that what the body really wants to, in terms of nutrients to be detecting is what is entering the body, not what is just sitting in the lumen, because if it's going to trigger insulin secretion and appetite, it wants to know really what's coming into the bloodstream at the time and not just what happens to be sitting in the gut lumen. So when I was giving this sort of talk years ago, I used to say that I thought the enterocrine cells were sitting there detecting what's in the gut lumen. And I think I've now changed my story. I don't think they're detecting what's in the gut lumen. I think they're detecting what is coming through the epithelium, because that's more physiologically relevant. And just in the last few minutes, I wanted to take you through to some unpublished data we've got in the colon, because nutrient absorption under most circumstances is mostly gone by the time we've got to the colon. So what on earth is happening in colonic L cells? And there are a lot of them, and we showed a few years ago that as well as producing GLP-1 and PYY, they also produce this hormone insulin-like 5. So here's co-staining of a colonic L cell for GLP-1, PYY, and insulin-like 5. We found insulin-like 5 was in about half of L cells in the colon and rectum, in the mouse and human. It's actually a pseudogene in several other species, so you can't do this in rat and dog. But mouse and human have insulin-like 5. We found in a collaboration with Takeda that if you inject insulin-like 5 in mice, it's orexigenic. So this is quite unusual because it's co-released with GLP-1 and PYY. Well, it comes out of the same cells as GLP-1 and PYY, but these two are anorexigenic, and this one is orexigenic. So this is a bit unusual. And we had great trouble when we first published on insulin-like 5 because the immunoassays were terrible. They were batch after batch would not give the same response, and we couldn't make too much out of the data. There was a suggestion that insulin-like 5 concentrations are actually elevated after severe calorie restriction, and possibly that they're reduced after feeding. Now, I don't know if really that is the case. If it is, it says that whereas GLP-1 and PYY concentration goes up when you eat, the insulin-like 5 is going down. If that is true, it suggests that the GLP-1 and PYY are coming out from higher up and the colon is behaving differently in response to food intake. But when we first got those data, we also then wondered whether cells producing insulin-like 5 would respond oppositely in the colon to the ones producing that didn't have the insulin-like 5. So what we've set up fairly recently, because of this problem of measuring insulin-like 5 by immunoassay, is to measure it instead by mass spec. And actually just by taking you can take sorted cells or digested cells or lysed cells or tissue culture supernatants, and we can measure in parallel in the same samples a number of different gut hormones by mass spec. So here now is parallel measurements from colonic cultures of GLP-1, PYY, and insulin-like 5 by mass spec from primary mouse colonic cultures. And actually, you can see this is a range of different stimuli we've added. GLP-1, PYY, and insulin-like 5 all come out in parallel. So they're responding to the same stimuli and coming out together. And we looked at the co-localization in L cells of GLP-1, insulin-like PYY, at the vesicular level. And here you can see the overlap. So these are different vesicles all within one cell. We looked at lots of different cells and actually found there's variability between the cells, but that most cells have at least half of the vesicles have dual labeled, at least half the vesicles are dual labeled or triple labeled, and some of them are single labeled. So we actually think that the cells that producing these peptides, if they respond to one ligand, they'll actually release a mixture of anything they have. So this is my final summary slide. I think we need to think about potential different functional roles of enteroendocrine cells along the GI tract. I wonder what's happening down in the colon and rectum, and I suspect these are responding to microbial products, bile acids, and if you do have any undigested nutrients reaching that area, they will respond to them. And I'm assuming that this is involved in gut health and interprandial control of appetite and motility. And I think when we're thinking of immediate postprandial responses, we need to be thinking of the absorbed nutrients triggering cells in the upper GI tract here. And the middle, so the ileum, I think, is responding to slowly absorbed nutrients, but this is the region I think that is being brought into play when you have the Roux-en-Y gastric bypass and the nutrient absorption doesn't happen here, it happens down here, in which case you get these very strong control of insulin secretion and appetite. So there's my summary. This is my group members. I run my group with Frank Ryman. He's my husband, and we run it as a combined group. And thanks to the MRC and Wellcome, who are our major funders, and thank you all for listening.